This lecture will be on receptors and cell responses to signaling. The material here is not presented in the traditional book and is being added at the request of Dr. Wu and Dr. Sears. In this lecture, I will expect you to learn to draw a pathway of cell signaling for both an extracellular and an intracellular receptor. Be able to draw a protein kinase cascade. That's not a specific one, but just one in general. Define CAMP, kinase, G protein, and ion channel. Define and differentiate between paracrine, synaptic, and endocrine signaling, being able to give an example of each one. Multicellular organisms need communication between cells in order to ensure that everything is working in harmony and in synchrony. So we will look at cell-to-cell communication in this lecture. Why do we need it? Well, because coordinated responses require a coordinating complex. So they require something that is going to be telling all the different cells what to do. And the cells themselves need to say what part they are doing to other cells. Different cells must also be specialized to signal or to respond or to receive a signal and then respond to other cells. And we're looking specifically at two types, well, generally in two types, endocrine signaling and nervous signaling. Endocrine signaling is going to be such as is done by hormones, where you have an endocrine cell releasing a hormone into the bloodstream that will then travel to different cells and give a certain response. Nervous signaling, which we'll call synaptic signaling, occurs when nerves either signal to each other or signal to other cells that something is going on, and they do so at the end of the neuron, which is called a synapse. So we're comparing these also with paracrine signaling. This is when local signals occur between different cells. Say, if you have multiple cells growing in your body and they're next to each other, they may signal that I am here, you are there, and basically signaling back and forth to let the other cells know what the cellular density is and whether they're touching each other or not. It's a good thing for cells to know if they're all alone or if they're part of a cellular layer. Paracrine signaling occurs in very localized areas. The secretions will only go as far as the next couple of cells. Again, nervous or synaptic signaling is going to occur specifically with neurons. So neurons are going to send signals, and the signals are sent at the end of a neuron, which is known as the synapse. So the synapse gives off these signals, and they are only received by the cell that is in contact with the synapse. This is synaptic signaling. And there is also endocrine or hormonal signaling, which is long distance and utilizes the blood vessels of the circulatory system to move the hormone from the endocrine cell to the target cell. This means that the signal will go through all blood vessels and come in contact with all cells that are in contact with blood vessels. So all cells. So hormones go to all cells, but only some of them will receive the signal and some of them will respond differently. And we'll finish that up at the end when we talk about steroid hormones. And the differences to look at here between these three are the distance between the cell that is releasing stuff and the target, the means of transportation, whether it's just in the intercellular medium, whether it's at a synapse, or whether it's through the blood, and the speed of transportation. Synapses, for example, are the fastest. Paracrine signaling is often very fast, and hormonal signaling can be slower but is longer term. So let's look at different types of receptors. So in order for a cell to respond to a signal, it has to have a receptor that will receive that signal. So we're going to also look specifically here about different polar signals, because there are polar signals and non-polar signals. Polar signals will have to bind to a receptor on the cell surface because they cannot get through the cell membrane, which has a non-polar interior. And this is some hormones are going to be polar, and neurotransmitters are almost always polar. So these have these cell membrane receptors and then use something called a signal transduction pathway. So this pathway starts at the reception when a signaling molecule will bind to a specific receptor. Now, this means the signaling molecule has to look very specific in order to be bound by a very specific receptor. There will then be transduction. Transduction is when the signal is passed on by a series of relay molecules. This is called the signal transduction pathway. We use STP as the shorthand for this. And we're going to look at different transduction pathways such as cyclic AMP and kinases. And then there is a response. This can be a cytosolic response that occurs in the cytosol by some change occurring in the cell fluid there, or it can be a nuclear response where what you have is a change in the transcription of certain genes which will affect a response. So, reception, transduction, response. 3 parts of how a cell responds to a signaling molecule. And this is specifically looking at polar signals. One type of signaling response pathway is going to be G protein coupled receptors, or GPCRs. And what happens here is a signaling molecule will bind to an activated G protein coupled receptor, which is coupled with a G protein. A G protein is specifically going to use GTP, which is similar to ATP, as its turn on, turn off. When it's turned on with GTP, it will move away from the GPCR and towards some other enzyme, and then it will activate that enzyme, causing a cellular response. Many signaling transduction pathways use GPCRs because these are on the surface and can bind to signaling molecules. Some receive things on what are called ion channels. And this is also specific to synaptic transmission because neurotransmitters occur in neurons at synapses and thus will be binding to something on the other side, often an ion channel. This ion channel, when it receives the signaling molecule, will allow some ions to go into the cell. So perhaps this is going to be something like a neuron getting a neurotransmitter from another neuron and allowing sodium ions into the cell. And we're going to see later how that can cause an action potential, which is an electrical signal within a neuron. You can also get acetylcholine binding to an ion channel in a muscle, which will then allow calcium in, which is going to cause a response. Well, it allows sodium in, which will then cause a response, which is for the cell to release calcium, and then that will make the muscle flex. And what happens with an ion channel is there will be a change in the voltage across the cell membrane. If you don't understand that quite yet, we will get to this in more detail when we cover the nervous system, because ion channels are frequently found in the nervous system. And this does, as I mentioned, allow for signal transmission between neurons and muscle contraction. Phosphorylation cascades are going to use something called kinases. And these kinases are going to play a little game called pass the phosphate, you could say. Kinase means that they're going to add ATP to something, put a phosphate on it, and then that will activate the next kinase. So what happens here is a kinase will activate a kinase, will activate a kinase, will activate a protein. And that kind of downstream effect allows kinases to activate different kinases and perhaps branch off the signaling pathway into multiple different pathways, or if only one protein is present at the end, that protein can be different in different cells, allowing for a different response at the end. As I mentioned, kinases are known for transferring phosphate ions. An example of this, something used in this, is simvastatin, which is a cholesterol drug and can also inhibit the MAPK pathway, which is a MAP kinase pathway in cancer cells, which makes simvastatin a moderately effective anti-cancer drug because it inhibits protein kinases. There is also cyclic AMP. Cyclic AMP is similar to ATP because both have the adenosine, but cyclic AMP, adenosine monophosphate instead of adenosine triphosphate. And it's also cyclized to make it, cyclical. And this acts as a secondary messenger. Now, what's going to make it cyclical is adenylyl cyclase. Adenylyl cyclase takes ATP, turns it into CAMP, which can activate further things, such as a protein kinase and therefore a phosphorylation cascade. This is very common if the signaling molecule is message outside the cell. CAMP may be the message inside the cell. And you see here as well, there is a G protein. That G protein is activating something. That something is adenylyl cyclase. The adenylyl cyclase causes CAMP to occur and causes that CAMP to activate a protein kinase, which activates a phosphorylation cascade. So we have 3 separate things going on here, and that's pretty common. Multiple different pathways can be part of the transduction. There we go. Signaling by steroid hormones. So steroid hormones are often nonpolar and can just go straight through the plasma membrane. When they do so, they don't need to bind to a surface receptor. So there aren't G proteins, ion channels, kinases, or cyclic AMP there. They just find the actual receptor in the cell. And when they bind with the receptor, that receptor can then do something in the cell. It may actually do something like going into the nucleus, and when it finds the DNA, transcribing some mRNA or enhancing the transcription of certain genes. So it can either be a transcription factor itself, or it can enhance transcription, or something like that. And this allows for a direct response of the nucleus. So there is reception, but the transduction doesn't happen so much as just a response. Well, we can have one hormone like epinephrine, and this hormone can have multiple different responses. For example, in a liver cell, epinephrine will bind to a beta receptor, causing glycogen deposits to break down into glucose and release energy from the cell. Epinephrine can also bind to a smooth muscle cell in the wall of blood vessels that supply skeletal muscles. And epinephrine therefore makes the blood vessel dilate, increasing blood flow to skeletal muscle. So you've increased glucose availability, you've increased blood flow to skeletal muscles. This is the same molecule, the same hormone, doing the same thing, or different things in different cells. But for both of these, that's going to increase the energy available and your ability to use it. This is good for fight or flight. Now, this is also epinephrine going to bind to alpha receptors in smooth muscle cells in the wall of blood vessels, but these ones that supply the intestines, causing these to contract, reducing blood flow to the intestines, so you're no longer resting and digesting, you're in fight or flight mode. So this fight or flight mode used by epinephrine has one hormone, multiple responses, including different responses if there are different receptors. So you can see how this signaling becomes very efficient if you can do different things in different cells. So when we mention cellular differentiation, this is why. This is also why we don't nurse babies and ovulate at the same time, because prolactin actually inhibits ovulation at the same time that it's pro-lactation. All cells have the same genome, and the expression of different parts of their genome is going to allow them to have different receptors, different transduction, and different responses.